Hey guys, so today we're moving away from photosynthesis and getting started with cellular respiration. So we're going to start by going over what the whole purpose of cellular respiration is, and we're also going to go over the first step, which is glycolysis. And then in the next lesson, we'll go through the rest of cellular respiration. So for this lesson on cellular respiration, we're going to be going over the reactants and products of cellular respiration. We're going to go over the different molecules that transfer electrons and undergo redox reactions. We're going to talk about how ATP is used in cells and how glucose is converted into ATP energy. We're going to briefly talk about the difference between aerobic and anaerobic cellular respiration, and then we're going to get into the first step of cellular respiration, which is glycolysis. So the chemical equation for cellular respiration is that we take glucose and oxygen, and it becomes carbon dioxide, water, and energy. Anytime cells require energy for different cellular processes, they can't actually use the glucose, which has potential energy stored in its bonds. Instead, we have to convert that stored potential energy into usable energy, and in cells, the usable energy is ATP. So the whole point of cellular respiration is to just convert the energy stored in glucose into ATP energy. Like in photosynthesis, there are going to be redox reactions occurring during cellular respiration where electrons are moved between molecules. Anytime you have electrons moving between two molecules, one of those molecules is losing the electron. That molecule is being oxidized. The other molecule is gaining the electron. It's being reduced. So together it is a redox reaction. NADH is an electron donor, which means it can give up an electron. So it becomes oxidized when it gives up an electron. NAD plus is an electron acceptor, so it gains an electron, which means it becomes reduced when it gains an electron. FADH2 is also an electron donor, so it will give up its electron in the process of oxidation. FAD plus is an electron acceptor, so it is reduced when it accepts an electron. So all of these molecules, the NADH and the FADH2, they transfer electrons via these different redox reactions where you have a reduction and an oxidation taking place. This transfer of electrons, these redox reactions, are going to release energy that can be used to make ATP. Just like in the electron transport chain of photosynthesis, when we had those redox reactions and it released energy to pump the hydrogen ions. It's going to be very similar in cellular respiration. Okay, why do we need this ATP energy? It's essentially to power a bunch of different functions in the cell. So things like how chromosomes move to different sides of the cell during cell division, active transport, so pumping molecules across the cell membrane, either into or out of the cell. An example of that is the sodium-potassium pump. Bioluminescence, so when some organisms are able to glow. Muscle contraction, so anytime you want to move, that's requiring muscle contraction. So it was like your heart beating, your diaphragm contracting as you breathe. Protein synthesis, so anytime your body is trying to make new proteins, whether it's so that you can make new skin cells or for your hair to grow or anything like that. DNA replication, again, which is important for making new cells during mitosis or meiosis, and switching on and off enzymes, among other things. A glucose molecule contains about 100 times as much energy as ATP, like a molecule of ATP. Glucose is obviously very high energy. It's also highly soluble, so it dissolves really well in water, which is what makes it such a good storage place for energy. It can hold a lot of energy and it can move in and around cells really easily because there's a lot of water in the cytoplasm and in the extracellular fluid. But again, glucose cannot be directly used by the cell. We have to turn that stored energy in the glucose into ATP first. And then just like in photosynthesis, in order to release the energy of ATP, you just break off that third phosphate. So we end up with ADP and an extra phosphate. And when you break off that phosphate, it releases the energy, and that's the energy that we can then use. So the main role of cellular respiration is just to transfer energy from food molecules like glucose into usable ATP energy. Only about 36% of the energy stored in glucose is actually converted into ATP. So that process of making ATP from glucose is only about 36% efficient. The remaining 64% is just lost as heat or thermal energy. In mammals and birds, that heat energy is actually still important. It's not like totally useless energy. It's how we maintain our body temperature, is with the released energy from cellular respiration. There are two different types of cellular respiration. The first is aerobic cellular respiration, and aerobic just means that it takes place in the presence of oxygen and it needs oxygen. This process of aerobic cellular respiration involves the complete oxidation of glucose, which means in the end, we have 6 carbon dioxide, 6 water, and 36 ATP. So this is where the 36% efficient comes in. If we're doing aerobic cellular respiration, it is 36% efficient. We will get 36 ATP from the glucose. And it does involve 4 stages, the first of which is glycolysis. We're going to talk about later this lesson. Anaerobic cellular respiration, on the other hand, takes place in the absence of oxygen, so there is no oxygen present. In this case, the glucose is not completely oxidized, so it's not completely broken down, essentially. There are still carbons that are not attached to oxygens, that are not made into carbon dioxide, essentially, is what it means when it's not completely oxidized. There are 2 different types of cellular respiration, and we'll talk about those 2 different types in a later lesson. But both types of anaerobic cellular respiration have only 2 stages instead of 4. Those stages all occur in the cytoplasm, and you'll see when we look at the aerobic cellular respiration later, they don't all take place in the cytoplasm. And perhaps most importantly, with anaerobic cellular respiration, both types, you only end up with 2 ATP being produced for every glucose molecule. So instead of being 36% efficient, the way aerobic cellular respiration is, anaerobic cellular respiration is only 2% efficient. So very, very low percent efficiency, which is why a lot of animals, like ourselves, have evolved to the more efficient method of aerobic cellular respiration. One of the types of anaerobic respiration produces ethanol and carbon dioxide. The other produces lactic acid. Again, we're going to talk more about anaerobic cellular respiration later, but it is important to know that it is only 2% efficient compared to aerobic, which is 36%. The other thing, though, with anaerobic cellular respiration is that it does have the same first step as aerobic cellular respiration. So whether it's anaerobic cellular respiration or aerobic cellular respiration, the first step is always glycolysis. So glycolysis is anaerobic. It's an anaerobic process. It does not need oxygen. This is why it can be the first stage of not only the aerobic respiration, but also the anaerobic respiration, because this step doesn't need oxygen. It does occur in the cytoplasm of the cell. Essentially, you take a glucose molecule, which is a ring of 6 carbons, and you snap it in half to make 2 pyruvate chains. Each pyruvate is going to have 3 carbons. So we took a 6 carbon ring, split it in half into 2 3 carbon chains. Those are called pyruvates. Glycolysis is a series of 10 reactions. You do not need to know all 10 of the steps. Each reaction is catalyzed by a specific enzyme. You also do not need to know the specific enzymes. During these reactions, you do actually need to put in 2 ATP in order to kind of kickstart this reaction. It's called an investment of energy. You're putting energy in order to get more energy out. So we break those 2 ATPs, break their phosphates off the end, that releases energy, allows us to power this glycolysis reaction. And then in the end, we get 4 ATP back, which means we have a net gain of 2 ATP. So we put 2 in, we get 4 back. We've made essentially 2 ATP. It would be like if you gave someone $2 and they gave you 4 back, you've just made $2. So these ATP are, of course, made by bonding a phosphate to an ADP. That's how we always make ATP. And then those ATP are going to be used to power cell functions. During glycolysis, we also have 2 NAD+ that come in. They are reduced because they're going to get some hydrogens and, more importantly, some electrons. So they're going to form 2 NADH molecules. You also have some hydrogen ions that are going to be floating around. The NADH are high energy. They are going to contain high energy electrons. Like we said, the original glucose contains 6 carbon. So each of the pyruvates are going to have 3 carbon. And then the NADH that is produced is going to be used later on in the 4th step of glycolysis. Okay, so this is kind of the net reaction here. We have our 1 glucose, the 2 ATP and P, so ADP plus P is just 2 ATP. So the glucose, the 2 ATP, and the 2 NAD+. From that, we're going to get the 2 pyruvate, which is where most of the energy is still going to be after glycolysis. We're also going to have 2 ATP, which has some energy, and the 2 NADH, which has some energy, and then the 2 hydrogen ions are just going to float away. Okay, this is just kind of another summary of what this looks like. So this is what our 6 carbon glucose looks like. We're going to put in the 2 ATP. We're going to get out 4 ATP. We're also going to put in the 2 NAD+. We are going to reduce them to become the 2 NADH. Our hydrogen ions are going to float away. And we also end up with our 2 pyruvates, which are the 3 carbon chains. Again, at the end of this glycolysis reaction, most of the energy that was in the glucose is still in the pyruvate. We actually haven't gotten a lot of energy out of it yet. And so the next 2 steps are going to be all about getting the remaining energy out of those pyruvates. So all organisms use glycolysis to make ATP. Again, whether they're anaerobic or aerobic, they all start with this glycolysis step. It is only about 2% efficient at converting energy from the glucose form into the ATP. Some of the energy is then released as heat, but most of the energy is still in that pyruvate. There's also some of the energy still in the NADH molecules. Remember, they're carrying high energy electrons, just like the NADPH in photosynthesis. They're very similar in how they function. The one in photosynthesis just has a P in its name. So these molecules have to be processed through the next 2 steps of aerobic cellular respiration. If we are anaerobic and we don't do those next 2 steps, then your process is only 2% efficient. The other 34% of the energy that we get in aerobic cellular respiration is going to come in the next 2 steps. Some single-celled organisms do only use glycolysis and they don't do the rest of cellular respiration. But for multi-celled organisms that need more energy, glycolysis alone just doesn't provide enough energy. It's not efficient enough, and that is why we have evolved aerobic cellular respiration. Okay, so I've just kind of drawn out aerobic cellular respiration here. This is step 1, glycolysis. That's what we were just talking about. The second step is right here. The third step is right here. And then the fourth step is over here. We're going to go through steps 2 to 4 next, but right now we have just done glycolysis. Again, it takes place in the cytoplasm. We start with our glucose, which has 6 carbons in it. It's going to turn into 2 pyruvates, which each have 3 carbons. In that process, we put in 2 ATP, we get out 4 ATP for a net gain of 2 ATP. We also put in 2 NAD+, which are reduced to become 2 NADH. Okay, and then we're going to continue on from there in the next lesson.